Did you know that Fragile X syndrome, or FXS, is the leading monogenic cause of autism? FXS is a neurodevelopmental disorder with dysregulated mRNA translation leading to phenotypic abnormalities like cognitive impairment, hyperactivity, anxiety, and seizures. FXS is caused by a genetic defect in the Fragile X messenger ribonucleoprotein 1, or FMR1 gene located on the X chromosome, and therefore it disproportionately affects males compared to females. Extracellular signal-related kinases, or ERK, are overexpressed in FXS, leading to abnormal mRNA translation, and S6 is a downstream effector of this pathway. Adjusting the levels of ERK could potentially correct the dysregulated mRNA translation and associated phenotypic abnormalities. However, an important question related to researchers remains unanswered. How do the levels of ERK activation change with development? Hi everyone, my name is Nidhi and I'm currently a member of the Cheng Lab. I'm working on an ongoing project to look at ERK activation levels during postnatal development in a mouse model of FXS and wild-type mice. First objective of this project explores the efficacy of the PD drug, a repressor of ERK, on the ERK pathway in the cortex of knockout FXS mouse models, including downstream targets like phosphorylated ERK, PERK, ERK, phosphorylated S6, PS6, and S6. The second objective of this ongoing project is to determine the levels of ERK activation in neonatal, juvenile, and adult female wild-type and FXS mice. This is a novel project because no previous studies have looked at the difference in expression in different developmental stages, and female mice are largely understudied. To test both objectives, first brain tissue was lysed using REPA buffer and protein concentrations were calculated using a Bradford assay. A Western blot was performed and membranes were incubated with the appropriate antibodies. Actin was used as a loading control. The sample size used for this project was 6. First, let's begin with our visual analysis. The darker the bands indicate stronger protein concentration. For the first part of the project, we looked at the effect of PD drug on male FXS mice. So visually we can see that ERK looks even in the vehicle and PD drug treated groups. There is no PERK in the PD drug treated samples. The levels of PS6 and S6 are quite even across the lanes. However, the lanes must be semi-quantified using imaging and tests performed to assess for statistical significance between the various groups. This statistical analysis is consistent with our visual analysis, and it shows that the PD drug repressed the levels of active ERK in the drug-treated samples. There was only a statistically significant increase in S6 levels for PD-treated group and no significance in PS6 levels, showing that PD drug does not influence S6 as initially hypothesized. Now let's move on to addressing the second objective of this project. The left side of the membrane is the wild-type samples and the right side are the FXS samples. From left to right, the samples are organized into neonatal, juvenile, and adult groups. Visually, we can see that there is much less sample in the juvenile group compared to neonatal and adult groups. From our results, it is seen that the levels of ERK activation are quite dynamic. From the first graph, there is a statistically significant difference between all ages in wild-type and FXS mice for PERK normalized by actin expression levels, except for FXS neonatal and juvenile mice. From graph 3, there seems to be a decrease in ERK activation in FXS in our neonatal mice compared to wild-type. ERK activation is increased in juvenile mice and significantly more in adult mice. There's also a statistically significant decrease of ERK activation levels from wild-type neonatal to juvenile, and then an increase in adult mice. Overall, we cannot generalize ERK activation levels in different developmental stages. This information provides direction for future therapeutic targets like the most effective brain region and developmental stage to administer PD drug for FXS mouse models. The next steps for this project are to continue to explore ERK activation levels in different brain regions and peripheral tissues such as the liver and heart. The other objective is to look at male mice and determine any differences between the two sexes. I would like to acknowledge HBI, ACRI, and the University of Calgary VetMed for providing me this opportunity. I would also like to thank Veronica for helping me with tissue collection, Song for his help with visualization and running gels, May for her help with the Western blot procedure, and Dr. Cheng for supervising